Let me start with something that I find genuinely fascinating, and that I think will reframe how you think about the food on your plate. Imagine two people sitting down to the same meal. Same portion of rice, same time of day, same general state of health on paper. One of them finishes eating and their blood sugar rises sharply, a spike that taxes the pancreas, stresses the blood vessel walls, and sets off a cascade of effects that will leave them feeling sluggish and hungry again within 90 minutes. The other person's blood sugar rises modestly and steadily. Their insulin response is measured and proportionate. Their energy stays stable through the afternoon. Their pancreas is not being pushed to its limits. Same rice, same quantity, completely different biological outcome. The difference is not the rice. The difference is what was eaten alongside it, and more specifically, how the other foods in that meal influenced the rate at which glucose entered the bloodstream, the efficiency with which insulin responded, and the degree of oxidative stress placed on the cells responsible for producing that insulin. This is how powerful food context is, and it is the central idea I want to explore with you today. Because if food combinations can protect your glucose response in that kind of way, if specific compounds in specific ingredients can slow glucose absorption, improve insulin sensitivity, and reduce the oxidative stress on your pancreas, then understanding which ingredients do this and how to use them is not a minor lifestyle detail. It is one of the most practical things you can do for your long-term metabolic health. Today, I want to walk you through 4 ingredients that are present in most people's kitchens right now. 4 ingredients with genuine, research-supported mechanisms relevant to blood sugar management, and explain exactly what is happening biologically when you use them, and how to use them correctly to get the best possible result. Before I do that, I need to say something clearly. This video is about nutritional support for blood sugar management. It is not about reversing or curing diabetes. Type 2 diabetes and prediabetes are serious medical conditions that require proper diagnosis, monitoring, and in many cases, medication. The ingredients I am about to describe are not substitutes for your doctor's care or your prescribed treatment. They are evidence-backed dietary tools that, used consistently alongside proper medical management, may support better blood sugar control over time. If you are on diabetes medication, please do not adjust your doses based on anything in this video without speaking to your doctor. Some of these ingredients do have real effects on glucose levels, and your doctor needs to know you are using them. With that said, let me first explain the biology, because once you understand what is happening inside your body when blood sugar rises, the mechanisms behind each ingredient become immediately clear. Every time you eat carbohydrates, rice, roti, fruit, bread, lentils, any starchy or sugary food, your digestive system breaks them down into glucose. That glucose enters the bloodstream. Your pancreas detects the rising blood sugar and releases insulin, a hormone that acts essentially as a key, unlocking the doors of your cells so that glucose can enter and be used as fuel. In a healthy, well-regulated system, this happens smoothly. Blood sugar rises modestly after a meal, insulin responds proportionately, glucose moves into cells efficiently, and blood sugar returns to its baseline within a couple of hours. The system handles it without drama. But the speed and height of the blood sugar rise matters enormously. When carbohydrates are broken down and absorbed rapidly, when glucose floods into the bloodstream in a sudden surge rather than a gradual flow, the pancreas has to release a large, rapid burst of insulin to manage it. Over time, this repeated high demand on the pancreas is one of the drivers of beta cell exhaustion, the gradual wearing out of the insulin-producing cells that are working too hard for too long. And in people who have developed insulin resistance, where cells have become less responsive to insulin's signal, so the pancreas has to produce more and more insulin to achieve the same effect, those blood sugar spikes stay elevated longer. The insulin response becomes increasingly disproportionate, and the cumulative damage to blood vessels, nerves, and organs accumulates silently over years. What the right dietary ingredients can do, and this is the mechanism I want you to hold in your mind throughout this video, is intervene at specific points in this process, Some can slow the rate at which carbohydrates are broken down into glucose. Some can improve the efficiency with which cells respond to insulin. Some can protect the pancreatic beta cells from the oxidative stress that damages them over time. And some can reduce the liver's contribution to blood sugar between meals. None of them work instantly. None of them work in isolation from the rest of the diet. But used consistently, as part of a thoughtful dietary approach alongside proper medical care, their contribution is real and measurable. Let us go through all four. The first is fenugreek seeds, known in Hindi as methi dana, and present in kitchens across South Asia, North Africa, and the Middle East for thousands of years. If I had to choose one ingredient that most clearly illustrates how a single food can intervene in the blood sugar process through multiple distinct mechanisms simultaneously, it would be fenugreek. Because fenugreek works through at least 3 separate pathways, and understanding all 3 explains why its effects are as consistent in the clinical research as they are. The first mechanism is physical. Fenugreek seeds are exceptionally rich in a specific type of soluble fiber called galactomannan. When you eat fenugreek, particularly when it has been soaked, which hydrates the fiber, the galactomannan absorbs water and forms a thick, viscous gel in your digestive tract. This gel does something elegantly simple. It physically slows the movement of food through your gut and creates a barrier that limits how quickly the digestive enzymes responsible for breaking down carbohydrates can access the starch molecules in your meal. The result is that glucose does not pour into the bloodstream all at once. It trickles in at a controlled, gradual rate. Instead of a sharp spike that demands a large insulin response, the blood sugar curve flattens. The pancreas responds proportionately rather than urgently. And the post-meal blood sugar elevation, the spike that causes long-term vascular and nerve damage when it happens repeatedly, is meaningfully reduced. Think of it as placing a speed governor on the rate of glucose absorption. The glucose still arrives. The carbohydrates are still digested, but the speed is controlled. The second mechanism is hormonal. Fenugreek contains a rare amino acid called 4-hydroxyisoleucine that is found in very few other foods in nature. Research on this compound has shown that it directly stimulates insulin secretion from pancreatic beta cells, but in a glucose-dependent manner. This is an important distinction. 4-hydroxyisoleucine does not force insulin release regardless of blood sugar levels, the way some pharmaceutical interventions do. It improves the insulin secretory response, specifically when blood sugar is elevated, making the pancreas more responsive at the moments it needs to be, rather than overstimulating it continuously. This means the pancreas is working more efficiently, producing more insulin when blood sugar is genuinely high, but not being overdriven between meals. Over time, this more precise, responsive behavior reduces the progressive exhaustion of beta cells that is one of the central features of type 2 diabetes progression. The third mechanism is gastrointestinal motility. Fenugreek slows gastric emptying, the rate at which food leaves the stomach and enters the small intestine. When the stomach empties more slowly, the entire digestive process is stretched over a longer window. Glucose absorption is spread out. Blood sugar rises more gradually after meals, and the satiety signals that tell the brain the meal is sufficient remain active for longer, which is why many people who use fenugreek consistently report feeling fuller for longer after meals and experiencing fewer energy crashes and cravings between eating. Multiple clinical studies have examined fenugreek's effects on blood sugar in people with type 2 diabetes. A study published in the European Journal of Clinical Pharmacology found that fenugreek seed powder significantly reduced both fasting blood glucose and post-meal glucose levels compared to placebo. A meta-analysis published in the Journal of Ethnopharmacology, pooling data from multiple controlled trials, found consistent and statistically significant reductions in HbA1c, the 3-month average blood sugar marker, with regular fenugreek use. How to use it correctly. The most effective and well-studied method is to soak 1 teaspoon of fenugreek seeds in water overnight and consume both the seeds and the soaking water first thing in the morning on an empty stomach. Overnight soaking activates the soluble fiber, softens the seeds for easier digestion, and appears to improve the bioavailability of the active compounds. If chewing whole seeds is difficult, they can be lightly crushed before soaking, or the soaked seeds blended into a small amount of warm water. Fenugreek can also be incorporated into cooking, added in small amounts to dal, vegetable dishes, or curries. The compounds are relatively heat-stable and survive light cooking well. Consistency matters more than quantity. 1 teaspoon daily, used consistently, produces meaningfully better outcomes than occasional large doses. Excessive amounts can cause digestive discomfort, particularly in people with sensitive stomachs, so starting small and building gradually is sensible. One safety note: fenugreek has real blood glucose lowering effects. If you are on diabetes medication, please monitor your blood sugar when you begin using it regularly and let your doctor know. The combined effect of medication and fenugreek may be stronger than your current medication alone, and your doctor may need to adjust your dose accordingly. The second ingredient is jamun seed powder, the powdered seeds of the Indian blackberry, Syzygium cumini. Most people know jamun as a seasonal fruit, enjoyed for its flavor and its characteristic deep purple stain. Fewer people know that the seed inside the fruit has been used in Ayurvedic medicine for centuries, specifically for blood sugar management, and that the research investigating why is producing findings that are genuinely interesting. The mechanism by which jamun seeds work is distinct from fenugreek's. Rather than slowing glucose absorption through fiber, jamun seed compounds work primarily at the enzymatic level. When you eat starchy foods, rice, roti, bread, potatoes, your digestive system uses specific enzymes to break those complex carbohydrates down into their component glucose molecules. The most important of these enzymes are alpha amylase, which begins the process in the saliva and continues in the small intestine, and alpha glucosidase, which completes the breakdown. The speed with which these enzymes work determines how rapidly starch is converted to glucose and absorbed. Jamun seed powder contains plant compounds, particularly ellagic acid, oleanolic acid, and jambolene, that have demonstrated inhibitory effects on both alpha amylase and alpha glucosidase in research settings. By partially inhibiting these enzymes, jamun seed slows the conversion of dietary starch into glucose. The carbohydrates in a meal are still digested, but the process is stretched over a longer window, producing a gentler blood sugar rise after eating. This is actually the same mechanism targeted by a class of pharmaceutical drugs called alpha glucosidase inhibitors, including acarbose, that are prescribed specifically for post-meal blood sugar management in type 2 diabetes. Jamun seed achieves a milder version of this effect through natural plant compounds rather than pharmaceutical synthesis. Beyond the digestive enzyme effect, jamun seed contains antioxidant compounds that research suggests may protect pancreatic beta cells from oxidative stress. High blood sugar generates reactive oxygen species, free radicals, that damage the very cells responsible for producing the insulin needed to manage that blood sugar. It is a damaging cycle. The antioxidants in jamun seed may help interrupt that cycle by reducing the oxidative burden on beta cells, allowing them to function more consistently over time. Some research also suggests that jamun seed compounds may improve glucose uptake by muscle tissue, the major site where glucose is cleared from the bloodstream after insulin signaling. Improved muscle glucose uptake means more efficient blood sugar clearance and less glucose remaining in circulation after meals. A study published in the Journal of Applied Pharmaceutical Science found that administration of jamun seed extract produced significant reductions in fasting blood glucose in diabetic animal models. Human studies are more limited, but show promising trends in post-meal blood sugar management with regular use. How to use it. 1/2 to 1 teaspoon of finely powdered jamun seed in lukewarm water, taken once daily after lunch. The post-meal timing is important. Taking it after eating allows the active compounds to interact directly with the digestive process of that meal's carbohydrates, rather than acting on an empty stomach. Start with the lower dose and assess digestive tolerance before increasing. Availability of quality jamun seed powder varies depending on where you are located. Look for products with no additives, no fillers, and a fine, consistent grind. Store it in a dry, airtight container away from moisture. The third ingredient is one that most people use as a spice without realizing the depth of its metabolic activity: cinnamon. I want to spend some time on cinnamon because the mechanism behind its effects on blood sugar is more specific and more biochemically detailed than most people appreciate, and because the variety you use matters considerably. The active compounds in cinnamon most relevant to blood sugar are cinnamaldehyde, cinnamic acid, and a group of polyphenols called proanthocyanidin type A polymers. These compounds work at multiple levels in the glucose management pathway. At the cellular level, the polyphenols in cinnamon improve what is called insulin receptor kinase activity. When insulin arrives at a cell, it binds to a receptor on the cell surface and triggers a cascade of signaling events that ultimately opens the cell to glucose uptake. One of the key enzymes in that cascade is insulin receptor kinase. And in people with insulin resistance, this enzyme's activity is reduced, meaning the insulin signal is received weakly and the cell does not respond efficiently. Cinnamon polyphenols enhance insulin receptor kinase activity, making cells more responsive to the insulin signal. They also reduce the activity of protein tyrosine phosphatase, an enzyme that normally dampens insulin signaling after it has been initiated. By improving the activation signal and reducing its inhibition simultaneously, cinnamon creates conditions in which cells respond more readily to insulin, clearing glucose from the bloodstream more efficiently. At the metabolic level, cinnamaldehyde activates an enzyme called AMPK, adenosine monophosphate activated protein kinase, inside muscle and liver cells. AMPK is sometimes called the body's metabolic master switch because its activation promotes glucose uptake by muscles and improves energy metabolism. When AMPK is active, muscles pull more glucose from the blood for use as fuel, reducing post-meal blood sugar spikes without placing additional demand on the pancreas to produce more insulin. At the hepatic level, in the liver, cinnamon compounds help reduce the activity of enzymes involved in gluconeogenesis, the process by which the liver produces and releases glucose between meals and overnight. In people with insulin resistance and type 2 diabetes, hepatic gluconeogenesis is often excessive. The liver releases too much glucose, even when blood sugar is already adequate. Cinnamon's inhibitory effect on gluconeogenic enzymes contributes to lower fasting blood sugar levels, the reading you take before breakfast, in people who use it consistently. And at the digestive level, like jamun seed, cinnamon inhibits alpha amylase and alpha glucosidase activity, slowing the breakdown of dietary starch into glucose. Clinical research on cinnamon and blood sugar has been mixed in its findings. Some trials show significant effects, others more modest ones. The variability is partly explained by the variety of cinnamon used. There are two main commercial forms: Ceylon cinnamon, sometimes called true cinnamon, from Sri Lanka, and cassia cinnamon, the darker, more strongly flavored variety that is most common in grocery stores. Cassia cinnamon contains meaningful amounts of a compound called coumarin, which can place stress on the liver at higher doses consumed daily over long periods. Ceylon cinnamon contains far less coumarin and is the variety recommended for regular therapeutic use. It has a lighter, more delicate flavor and is widely available at health food stores and specialty spice suppliers. How to use it: approximately 1/4 to half a teaspoon of Ceylon cinnamon daily, added to warm water, herbal tea, dal, vegetable dishes, or porridge. Gentle heating during cooking helps release the polyphenols. The key is consistent daily use at modest amounts rather than large occasional doses. Do not exceed 1 teaspoon daily of cinnamon in any form without medical guidance. As with fenugreek, if you are on diabetes medication, particularly insulin or insulin-stimulating drugs. Monitor your blood sugar when you add cinnamon regularly to your diet and inform your doctor. The fourth ingredient is one that appears in daily cooking across South and Southeast Asia so routinely that its metabolic significance is almost entirely overlooked. Curry leaves, Murraya conigii. These aromatic leaves, added to tempering, to dal, to curries, to chutneys, contain a range of active compounds with genuinely interesting research behind them for blood sugar and pancreatic health. The primary active compounds in curry leaves relevant to blood sugar are a class of carbazole alkaloids, including mahanimbine, girinimbine, and murrayinine, alongside quercetin, beta-carotene, and a range of natural flavonoids. The carbazole alkaloids have been studied for their effects on insulin signaling at the cellular level. Research suggests they improve the responsiveness of cells to insulin, addressing insulin resistance at its cellular root by supporting the signaling pathway that allows glucose to move from the bloodstream into cells. In animal studies, curry leaf extracts have consistently shown blood glucose lowering effects and improvements in insulin sensitivity. The antioxidant compounds in curry leaves, particularly quercetin and beta-carotene, provide direct protective effects to pancreatic beta cells. As I described earlier, chronic high blood sugar generates oxidative stress that progressively damages the beta cells responsible for insulin production. By providing antioxidant protection to these cells, curry leaves may help slow this damage, supporting more consistent insulin production over time. Curry leaves also contain fiber and plant compounds that exert mild inhibitory effects on carbohydrate-digesting enzymes, slowing glucose absorption from meals, in a mechanism that parallels what we saw with jamun seed and cinnamon. The effect is gentler than the other ingredients, but contributes to the overall picture of metabolic support. There is also research suggesting that certain compounds in curry leaves support liver health, specifically by reducing triglyceride accumulation in hepatic tissue. This matters because fatty liver disease and insulin resistance are closely linked. They tend to develop together and worsen each other. Ingredients that support liver health may therefore contribute to improved insulin sensitivity through this indirect pathway. A study published in the Journal of Plant Foods for Human Nutrition found that curry leaf extract significantly reduced blood glucose levels and improved lipid profiles in diabetic animal models. Human research is more limited, but the mechanistic evidence is consistent with observed effects. How to use them. The simplest approach is to chew 5 to 7 fresh curry leaves on an empty stomach in the morning. This is the traditional method used in Ayurvedic practice and allows the active compounds to be absorbed without the interference of food. Alternatively, and for most people more palatably, add curry leaves generously to your daily cooking. Light cooking releases their beneficial compounds while maintaining digestibility. They survive tempering in oil well and contribute both flavor and nutritional value to any dish they are added to. Fresh curry leaves are most potent. Dried curry leaves retain some activity, but significantly less than fresh. If you have access to fresh leaves regularly, use them fresh. Consistency is what produces results. 5 to 7 leaves daily, every day, produces meaningfully better outcomes than occasional large additions. Let me now bring all 4 together, because the way these ingredients complement each other mechanistically is as important as what each one does individually. Fenugreek seeds work primarily through soluble fiber, slowing the physical movement of food and glucose absorption, and through 4-hydroxyisoleucine, which improves the precision and efficiency of the pancreatic insulin response. Jamun seed powder works primarily through enzyme inhibition, slowing the conversion of dietary starch to glucose at the digestive level, and through antioxidant protection of beta cells. Cinnamon works through cellular insulin signaling, improving how cells respond to insulin at the receptor level, through AMPK activation in muscle tissue, improving glucose uptake, and through hepatic gluconeogenesis inhibition, reducing the liver's contribution to fasting blood sugar. Curry leaves work through carbazole alkaloid-mediated improvement in cellular insulin sensitivity, antioxidant protection of beta cells, and liver health support. These are 4 distinct mechanisms targeting 4 different points in the blood sugar regulation system. Used together consistently, they address the picture more comprehensively than any single ingredient could alone. The practical daily routine looks like this: soaked fenugreek seeds with the soaking water first thing in the morning, 5 to 7 fresh curry leaves chewed alongside or added generously to the morning meal, a 1/4 teaspoon of Ceylon cinnamon added to tea, warm water, or a cooked dish at any point in the day, 1/2 teaspoon of jamun seed powder in lukewarm water after lunch. None of these require significant time or expense. None require sourcing exotic ingredients. All 4 are present in most South Asian kitchens already, or are easily available at any grocery store or spice market. The honest timeline for results is this: within the first 1 to 2 weeks, most people notice improvements in post-meal energy stability, fewer crashes, less urgency around cravings between meals. Within 4 to 6 weeks of consistent use, some people begin to notice improvements in their post-meal blood sugar readings when monitored. Over 2 to 3 months of sustained consistency, fasting blood sugar and HbA1c may begin to trend in a more favorable direction. These are not guaranteed outcomes. Individual response varies based on the severity of insulin resistance, the overall diet, physical activity, and other health factors. What I can say with confidence is that the mechanisms are real, the research is supportive, and the safety profile of all 4 ingredients at the doses described is well established. Your blood sugar is not something that happens to you independently of your choices. It is a system that responds, continuously, every day, to what you feed it. The ingredients I have described today are not magic. They are specific nutritional inputs that support specific biological mechanisms. Used consistently, alongside proper medical care, a thoughtful overall diet, and regular physical activity, they contribute to the kind of daily metabolic environment in which blood sugar management becomes progressively easier rather than progressively harder. If this was useful to you, please subscribe to the channel and share it with someone in your life who is managing blood sugar and looking for evidence-backed nutritional tools to support their care. Leave a comment below telling me which of these 4 ingredients you are going to start with, and whether you are already using any of them. I read every comment, and your responses shape what I cover next. I am Dr. Julian Cross. Take care of yourselves, and I will see you in the next one.